that's anchored in the membrane, and it has the two domains that form the cleft, and then there's the beta-2 microglobulin that binds to it. Okay? And it presents, it holds antigens that are exogenous or endogenous. MHC1. Endogenous. And presents those antigens to what type of T cells? CD8s, cytotoxic T cells. And MHC1 is on all nucleated cells. Okay? MHC2, in contrast, has two peptides, and they're both anchored in the membrane, right? So they both have a domain that helps form the two bananas on a plate, cleft. MHC2 are present on the surface of antigen-presenting cells, correct? The professional antigen-presenting cells, B, dendritic cells, and macrophages. And they present what type of antigen? Exogenous to CD4s, right? So that's a quick review on what we talked about over the last couple of weeks. MHCs, major histocompatibility complex. That term itself really refers to this sequence of genes. That's the major histocompatibility complex. It's on human chromosome 6. And it's the human lymphocyte antigens, or human leukocyte antigens, human leukocyte antigens. Let's not worry about the mouse down here. We know more about the mouse, but let's concentrate on the human. Okay. One species is enough. 10-year anniversary from high school, 10-year reunion from high school. My husband and I sat next to the guy I dated in high school. The guy I dated in high school is a veterinarian. He took over his father's practice in the town where I grew up. And he started teasing my husband about being a specialist. My husband is a family physician. Birth, newborn, all the way to geriatrics. My husband was getting on his ear. He says, I'm not a specialist, I do everything. And my high school boyfriend said, you only deal with one species. Oh, that was a good dig. So, yeah, we know more about mouse, but let's just deal with the one species, okay? Well, I really got him good on that. Okay, human chromosome 6. If you notice, there's different clusters of genes here, and there's several of them in this complex. Okay. Some of them code for class 1, the HLA, A, B, and C, the ones in red over here, code for the class 1 peptides. Then there's some dispersed in here that code for class 2. They include these D, these HLA-D, shown in yellow. Can you see there's quite a few of them? Okay. And then there's class 3. Well, we'll talk about class 3 later. Class 3 is kind of the hodgepodge of other genes that are present in this complex. Okay, but HLA-1 is coded for by MHC1 coded for by HLA, A, B, and C, and then class 2 by all these D, HLA-D genes. Here's a more detailed rendition of this complex on human chromosome 6. One thing you have to understand about these genes and the proteins for which they code, there's a lot of variety. That's what the term polymorphism means. There's a lot of variety here in the MHC1 and 2 proteins that are expressed on an individual's cells. there's a large number of different genes in that complex. And every gene that's inherited, and you inherit, you know, 2 alleles of that gene, of that complex, 2 alleles of that chromosome from both parents. Every gene you inherit is expressed. They are all codominant. Okay, so that's going to lead to a lot of variety. And I want to go through that and reiterate that. MHC1, HLA, A, B, and C on nucleated cells. MHC2, all the Ds, all the HLA Ds, and they are on professional antigen-presenting cells. MHC3s are oftentimes referred to as the immune response genes. That's what that I are. Immune response genes. And there's just a hodgepodge of different proteins that are coded for in that complex. There's separate genes for different proteins that are involved in the immune response. Okay, so let's concentrate on the MHC1 and 2. Polymorphism, okay, reiterating this. Variety of genes. You can see here's the 3 different MHC1s, and here's all of them that code for MHC class 2. And you can see the variety, you know, the propensity of the ones that are inherited. You most humans have 6 different MHC1 molecules on their surface. And that's because you have HLA A, B, and C genes, and you inherit an A from your mother, an A from your father, a B from your mother, and a B from your father, and so forth. Okay, so you essentially have 6 HLA. And there's so many varieties of those genes that unless, I could say something really tacky, I won't, unless your parents are closely related, the chances are you will not inherit the same gene from both of them. Does that make sense? I was going to talk about a certain region and I dropped that. I better not. Okay. So there's a large number of genes that you can inherit, and you inherit 1 from each parent. Okay? So you usually have about 6 different MHC1s. The 6 that you have, they're codominant. They're going to be expressed on the surface. Okay, and that really comes down to that MHC, that alpha peptide. Remember, the MHC1, you have that 1 peptide, and then the beta microglobulin binds to it. So you're going to have 6 different varieties of that, typical human. When it comes to type 2, you have 8 or at least 6, because some people don't inherit all the varieties, but you have typically 8, 4 from each parent. And there's a lot of diversity in those Ds. Okay, so you typically have 6 to 8 different MHC2s. Now, the MHC2s, 2 peptides, and both of them are anchored in the membrane, right? And they both are involved with forming the cleft. You can have a mix and a match. Okay? Those peptides can rearrange in different varieties. Does that make sense? You can have different arrangements of those MHCs that you inherit. Okay? So, is that going to change the cleft? Yes. That's going to change the appearance of that cleft and what it can hold. And some people have extra HLA-Ds, so they have extra MHC2s. Just a lot of variety. Most people have 2 alleles for each gene. The combination results in great diversity expressed within a person and within the population. And something I'll bring up in the Triple T lecture, tolerance, tumors, and transplants. It's important with transplants. If you need a tissue transplant, they really try to match a few of your MHCs, there's not much hope that you can match all of them, but there's certain ones that they try to match, because in order to have a perfect match of your MHCs, either you have to have an identical twin, or you have to, there'll be one person, if you don't have an identical twin, there'll be one person out of 20 million that has the same MHC expression on their cells as you. And keep in mind, the MHC1 is on all nucleated cells. Right? Does that make sense? So they really need to match those MHC1s. Those are the critical ones. Does that make sense? Okay. There's a lot of diversity in the genes because there were some duplication of genes and then mutations in those duplicated genes that were maintained on the chromosome. So they think that's how we arrived at having such a diverse number of genes available. Okay, I think I brought home that. The big variations that you see in the MHC peptides are present in that cleft region, the domains involved in forming that cleft. And this shows in MHC1, you can see both of the domains are in that alpha peptide. And you can see the points, the areas that are highlighted in red are the amino acids that have the greatest variation. Those are all in that cleft area. Are they going to come in contact with the peptide that's bound there? Yes. Could these come in contact with the T-cell receptor? Yes. These in the alpha helix can come in contact with the T-cell receptor, and those and the ones here on the plate on the beta strands are going to come into contact with the peptide. Here's the case for the beta. Excuse me, for the MHC2. And you can see the major differences are in that beta strand. And again, it's on the beta strands, it's on the beta peptide, on the beta strands and on that alpha helix. So it's going to be areas that come into contact with the peptide and some of them with the T-cell receptor. Now, one thing I haven't really emphasized yet, and granted, I know as we go over the next few lectures, I'm going to be repeating things, but repetition always helps me remember things. So I'm going to go ahead and repeat things, but I'm adding on to that information. And this is one key thing. Each MHC cleft can only bind a certain set of peptides because of the amino acid sequences that are present in that cleft area. The amino acids that are in these key locations will determine which peptides can bind that cleft. If you have a lot of hydrophobic amino acids here, then that peptide needs to have some hydrophobic amino acids here. Does that make sense? If there's some that have charges here, then that peptide needs to have the opposite charge there. Does that make sense? And the peptide needs to have a complementary shape. So not every MHC1 can hold every endogenous peptide to which it's exposed. It will only bind a certain set. Does that make sense, I hope? You know, and there may be 2 or 3 that can bind to a certain MHC1 protein. Okay? Same holds true for MHC2. Only certain peptides from those exogenous antigens, a limited number are going to be able to bind to an individual MHC2. So it really comes down to a semi-specific attachment. Okay? Granted, with 2, you can have varying lengths of peptides attached, so that gives some variety there. Okay? But MHC1 can't bind to every endogenous peptide. Boy, that'd be a great true-false statement next time.
What was that?
Say that again.
MHC cleft on an MHC1 cleft can bind any endogenous peptide. That's false. That's definitely false. Of course, now